And Dr. Kelman, why don't you tell us all about the reference materials?
Be happy to. I'm going to share my screen because I do have slides to show you, I hope. Oops, would be helpful if they were a little larger. So I'm going to talk about the NISTmAb, the NIST monoclonal antibody, and the NIST CHO, CHO cell line. These are reference materials for use by the biopharmaceutical industry. And I have my name on the slide, but my husband, Zvi Kelman, who's also here, will be assisting. I do need to give a disclaimer. Any opinion that Zvika expresses today is his own. He is not here representing NIST or the United States government. Zvika and I met when we were both in graduate school at what is now Weill Cornell Medical College. And he was working at the time on DNA replication. And so he is a protein guy as well as a molecular biologist. I'm really more of a molecular biologist. I was working on histone gene regulation. And therefore, when I talk about proteins, I always say that I'm faking it. So I'm quite sure I will say something incorrect during this talk. And hopefully Zvika or somebody will correct me. So this is a brief outline. We're going to talk about what NIST is and what reference materials are, a little bit about monoclonal antibodies, then specifically NISTmAb, which is a reference material sort of for the downstream processes since it's an antibody, and NIST CHO, which is a cell line that can serve as a reference material for upstream processes. I do hope you'll have plenty of time for questions, but I really want to stress that please, if you have a question, feel free to interrupt. And if somebody puts a question in the chat and somebody else says, you know what, this should be asked, just turn it on. I'm not so sure I'm going to be able to see the chat while I've got my screen shared. So please interrupt us with questions. So first of all, a little bit about NIST. This is the National Institute for Standards and Technology. It is a government agency that falls under the United States Department of Commerce. And when I tell you a little bit more about the standards that they set, I think that association with commerce will make sense to you. NIST was founded in 1901. And until 1988, it was called the National Bureau of Standards. And then it moved out of Washington, D.C. to its current, there are several locations, but one of them is a 550-acre campus in Gaithersburg, Maryland, which is where I live. And the roots of NIST actually go back to the United States Constitution, Article 1, Section 8, where it states that the government will, quote, fix the standards of weights and measures. And so the mission of NIST is very important. There's a lot of research that goes on there besides standard setting. 5 Nobel Prizes were awarded to scientists who performed their research while at NIST. But one of the important things about NIST, or something they're rather proud of, is it's a non-regulatory agency. So unlike the FDA, for example, which regulates food and drugs, NIST is purely in for lack of a better term, an advisory position. And what I've heard from NIST people, oh, and my lights have gone out, but I'm just going to keep talking. What I've heard from NIST people is that often makes the conversations between NIST and, for example, the biopharmaceutical industry, there's more freedom to speak to people. This is a quick aside, but just in case you didn't know, NIST has a summer undergraduate research fellowship, the NIST CERF program. These are competitive undergraduate fellowships held, I think they have them at Gaithersburg and also Boulder, Colorado, and possibly another of their sites. If your students are really, really good, you might have them check this out and possibly participate in it. So back to the talk. Well, one of the questions is, why do we need standards? And hopefully I don't have to convince you too much but things like length or distance, time, weights or masses, these things should be consistent. And there's a good chance you already know about the standard kilogram, the standard second, different standard units. But another thing besides that is that things like electrical components should be compatible with one another. So if we didn't have standards set for the idea that plugs should all fit into United States electrical plugs and should operate on 120 AC voltage, etc., we'd have a lot of problems. One of NIST's first big projects was after the Great Baltimore Fire of 1904, which burned most of Baltimore to the ground. And one of the problems in calling in different fire companies was that the hose connections, for example, were not standardized at the time. And so you couldn't do what you can do today, which is hook up to any fire hydrant or have compatible components between different fire trucks. So safety and electrical safety, fire safety, building safety, these have all been parts of NIST's mission from the beginning. And again, I'm hoping you're thinking, that's why it sort of falls under commerce, state to state, things like that. Also, in terms of standards, your building materials should have strength. You should be able to describe the quality. If you say that a building standard is that your steel has to be so strong, somebody needs to be able to prove that their steel is so strong. And we also need industry-wide comparisons that should be possible. So hopefully I've convinced you that standards can be a good thing. And then again, to go slightly off topic, about 9 years ago, Reddit absolutely blew up with the NIST peanut butter thing. Zvi mentioned last night that I think it's still available, but it's possible that you can no longer get SRM 2387, standard reference material, peanut butter. NIST has, I believe, thousands of different standard reference materials and reference materials, and some of them might not be what you're thinking. For example, the peanut butter. So if you're a food manufacturer and you want to report nutritional information, you need to calibrate your instruments so that when you say, hey, our peanut butter or a food that is in some way similar to peanut butter has this much fat and this much protein, then you need a reference material in order to check that your instruments are working correctly and the data and results that you determine could be reproduced. You could have a lot of fun checking out on Reddit back when they said that, and at the time, I believe this SRM cost about $700. So of course, people were going crazy over who would pay $700 for a bottle of peanut butter. But the answer is the industry will, because they need it. Please don't buy NIST peanut butter. It may or may not taste good. Some other standards provided by NIST are NIST standard cigarettes. These are not for people to smoke, but rather they're used in flammability studies. This is a standard way to test flammability of furniture or clothing, and it's a repeatable way for people to start a fire. Upper right corner, if you need human urine with or without marijuana metabolites, NIST's gotcha. They have several different human urine freeze-dried samples, which you can use to make sure that your drug testing equipment is functioning properly, for example. The lower left is one of the many different human things that we test for in like blood tests. So again, you can get something reproducible that you can make sure your equipment's working properly. And on the lower right are some of the various NIST standard concrete mixtures, which I hear from NIST people is one of their most popular best-selling reference materials. So NIST is there helping many different industries across the United States to do a better job of what they're doing. And just because I said US, I don't believe NIST prevents anyone from outside the US from using NIST materials, but they are, of course, produced for the United States. So this brings us to the life sciences and the question of, well, why would you need a reference antibody? And one of the reasons is that, well, in the biopharmaceutical industry, you need to calibrate and validate your instruments. If you're making, for example, a monoclonal antibody to be injected into humans, well, you really had better be sure that you're characterizing that correctly, that your quality control processes are repeatable and traceable to something. And so just some of the characterizations that somebody making a monoclonal antibody might care about could be the protein concentration, the primary sequence of the heavy and light chains, post-translational modifications. Some of the ways you could do this would be what that antibody looks like on an SDS polyacrylamide gel or using CE, capillary electrophoresis. You might be interested in what your protein looks like intact and fragmented using different mass spectrometry methods or NMR, structural determination. You might care about the aggregation state, whether there are particulates, whether that's coming from the protein itself or something else. These are just some of the things that someone might care about. The idea of having a standard antibody or a reference antibody where you could compare, for example, an instrument measuring something on that standard antibody, this allows an apples to apples comparison. Now, there's a very important point on this slide, and it's the asterisk after concentration, because Zvi will tell you, or remind me to tell you if I had forgotten, that when we talk about the NISTmAb, which will be the reference antibody, NIST actually only guarantees one thing about it, and that is the protein concentration. So actually using NISTmAb for anything else is they're not going to stop you from doing it, but NIST, as I'll show you, they characterize this extensively, but there's only one thing that they will tell you is repeatable, and they're very sure about it. So I know that this hackathon is about monoclonal or antibodies, and chances are you know plenty about them, but I just did put in 2 slides, and I will go through them quickly, and truly, if anyone has a question, please ask. So one way to think of monoclonal antibody structure is shown here, where there are 2 heavy chains, the longer polypeptides, 2 light chains, the shorter polypeptides, and that forms a heterotetramer with the what we love to call Y-shaped structure. The top portion of the molecule, often called the FAB fraction, that in terms of function is where antigen binding occurs. The lower portion of the molecule, the so-called FC fraction, that is, it is both involved in immunity, but the FC fraction, and I'm sorry, I have a different, whoops. I'm hoping people can hear me because my computer just froze. There it goes. The FC region of an antibody is species specific. And so the FC region from a human antibody identifies that antibody as being human in origin if we looked in a mouse, the FC region would identify it as a mouse antibody. If it were a goat, FC would be goat. And as you probably know, this is also, the FC region is also antibody class specific. When making a drug and knowing that the FC region is species specific, if we're going to put our drug into humans, if we, for example, made an antibody in goats, and this was a goat antibody against whatever the target is, and the FC region described it was goat, then if you gave that to a human, the human would mount an immune response against the goat FC region. And the antibody might or might not function, but if you were to ever give the human another dose of a goat antibody, they would have a fast immune reaction to it. That antibody would not be, would not give the right effect, probably would actually give a bad effect. So in the industry, when making an antibody, and this, the source, the initial source of most, I'm going to say all, but I don't honestly know this, the initial source of all monoclonal antibodies used as human therapeutics is probably not a human. Using mice, for example, is very popular. So we can't put a completely mouse antibody into humans because they're going to have this anti-mouse FC response. So you'll see therapeutic antibodies for humans typically described as human, humanized, or fully human. And these are different ways to change the FC region so that you can use it as a therapeutic in humans, and the humans do not mount, theoretically, an antibody response. Now, a human antibody, that's made starting in human cells. The FC region is human. A humanized antibody can be made beginning in a variety of non-human cells. The FC region is going to look human enough to not be particularly antigenic. And that can happen either by engineering the cell or the organism to already have human-ish or humanized antibodies being made by its immune system, or this can be engineered in at the DNA level during the monoclonal antibody producing cell line production. Fully human antibodies, similarly, can be made starting in a variety of non-human cells. In this case, the FC region is fully human, again, not particularly antigenic, but it didn't start in a human cell. So that's the difference between, and again, somebody can probably give a better description of this, but that's sort of the difference between different types of monoclonal antibodies made to be used as therapeutics in humans, and what you have to do to them so that the humans don't mount a response against the antibody. In thinking of monoclonal antibodies, another way to think about them is that they don't only have polypeptide chains, but they have a number of post-translational modifications. They do often have N and C terminal modifications. They have a number of disulfide bridges. And I should just mention that every antibody diagram you're going to see in this talk is just a generic diagram taken off the web. So the number of disulfide bridges will vary in different antibodies. And antibodies, as you probably know, are often glycosylated, not shown very well in this particular image. And they're typically a large number of different glycoforms associated with one monoclonal antibody. And what that does is, because these are large molecules, it makes them difficult to characterize, difficult to work with. I'm probably telling you things you already know. However, NIST developed reference material 8671, the NISTmAb, a reference antibody. And the way that they did this is NIST scientists did not produce this antibody, but a biotech company did. MedImmune, before it was acquired or merged with AstraZeneca, had produced an anti-RSV antibody, and they had gone through a large-scale production. However, that particular antibody did not go through the full FDA approval process. I'm not completely sure what happened. The rumor is that the antibody worked in humans against RSV, but it did not work better than an already approved antibody. So basically, MedImmune had a very large amount of what they donated to NIST and became known as the NISTmAb. Okay, so NIST then produced this as a reference material. So if you get some NISTmAb, you will get in one vial, you'll get 800 microliters. It is a humanized IgG1 kappa antibody. The concentration is 10 mg per mil, and it's in a histidine buffer at pH 6. The antibody as provided had already been formulated. I do believe NIST diluted it, but it was really in a form where it could have been used in humans, although nobody expects anybody to use NISTmAb in humans. It's not for that purpose. The epitope against which the NISTmAb was generated has been published, and the peptide sequence is shown on this slide. The extinction coefficient is 1.42, and the molecular weight is that of a typical antibody, somewhere around 150 kilodaltons. And Zvika, did you happen to look up? You have a slightly better number, if I recall correctly.
I think it's 158, but I'm not sure.
Okay, it may be published. I'm sure it's published. Yeah.
I just didn't ask.
Yeah, but again, as is the case with any antibody, that is not a single number, that there's going to be a spread around that, due mostly to glycosylation differences. So that NISTmAb has a humanized FC region that it can be protein A purified by affinity chromatography. And I'm sorry, the way I wrote this on the slide, you don't have to be humanized to be able to be protein A purified, but it happens to be humanized. That FAB region is going to bind that RSV epitope. One of the issues around community college use of, for the moment, I'm going to say NISTmAb, although I'm going to tell you in a moment, you probably do not want to actually order any NISTmAb because it's very expensive. But there is no readily available expression cassette for that epitope. Sandy, please ask.
Okay, Lori, where does protein A bind?
Oh, that's a good question. I don't know, somewhere in the FC region.
But you could purify FAB. I thought you could purify FAB fragments with it.
So I have a feeling it binds up even above the hinge. This is complete hand waving, Sandy.
Wait.
Go ahead.
You can purify the Fab, but it's not with protein A. CH1 column will bind to the FAB fragment.
Oh.
Yeah, I was about to say that. There is a resin that will bind the CH1 region. Personally, it's more expensive than protein A, and protein A is already fairly expensive. So depending on what you want to do, if you're making an intact antibody, protein A, which hand waving binds the FC region, that's the way to go. If for some reason, here's a reason to do it, if you want to monitor how much FC versus FAB you've got in a production run, for example, you can use these different binding resins to explore that. I'm not sure you'd want to do that in a community college biomanufacturing course, but you could. And I could get you a better answer to that if you
Oh,
wanted.
no, that's good. I just
Yep.
kind of, I always had the idea bound to the FC region, but thought I recalled that we might be able to use it to purify FAB fragments. So that's why I was wondering.
So that I actually know the answer to, because I did a sabbatical, and specifically using that anti-CH1, you pull FAB, but not intact. I said that wrong. They bind different things.
Okay.
So,
Now,
yeah.
and just to clarify, proteinase ones are protein G, protein L. Each of them bind slightly different location, but protein A is not the only ones that you can use for purification.
Yeah, and it looks like Todd's on Google, so thank you, Todd.
Thanks.
So while we know the epitope, at this moment in time, I am not aware of anybody selling, for example, a ready-made expression cassette or expression plasmid to ask some other to ask some kind of cell or bacteria to make the epitope. And there is no ELISA kit available for this. And I know that we have the question, which Zvika, you might want to wait until the end, of did NIST or does NIST have a functional test for this antibody? Do you want to say that now, or do you want to wait till later?
Well, we have binding assay. You saw the epitope, so we can bind the antibody to the epitope using either the BIA core or the octet. And then the FC can bind to the receptor, so we can do that assay as well. We don't have an assay for how effective it is against RSV, or effective it is to as inhibitory antibodies. So we are using routinely the binding either for the FC or for the epitope.
Okay, but you made, if I recall correctly, you had the epitope, you had the peptide made?
That's correct.
And it binds that.
Yes.
So that's a good thing. One of the goals of the NISTCHO hackathon team is to propose undergraduate research in developing an ELISA. So that will be a welcome thing. And hopefully, from what we just heard, it'll also be a doable thing, which would be great. Okay. The NISTmAb has been intensely characterized. There is a 3-volume American Chemical Society book series. These are the titles. I'm not going to talk about it, but everybody has a copy of the slides, or you'll get a copy of the slides in case you care. However, in case you were thinking, wow, I want some NISTmAb, I'm probably going to stop you now. So as is the case with the $700 peanut butter, NISTmAb is available for purchase, but one vial is almost $1,000. Now, just by the way, NIST does not make a profit on any of their reference materials or standard reference materials. They charge based upon a cost recovery model. So what you're really paying for here is an extensive amount of characterization and reproducibility. And, you know, again, you don't want to buy the NIST peanut butter, and chances are you're not going to purchase NISTmAb. So I'm sorry if I got your hopes up on that. But once the NISTmAb was released, and by the way, I have heard anecdotally, it's one of the most popular reference materials being sold, and a lot of instrument manufacturers actually provide some NISTmAb along with their equipment so that you can make sure it's performing properly. Industry immediately almost started asking NIST, hey, the NISTmAb is great. Can we get the cell line? And the answer is no. So first of all, just the question, what cell line made the NISTmAb? Well, we know that it was made by MedImmune. We know that it was made in NS0 cells, which is a murine or mouse cell line that's really good at expressing antibodies. However, that's really all we know. Cell lines are really proprietary. Companies do not give away cell lines. I'm going to say we're never going to know the actual specifics about the cell line that made the NISTmAb, but at least we won't know in the near or long-term future. So NIST thinking, well, wanted to help with this. And so Zvika's lab at NIST produced an E. coli strain that had an expression cassette with a DNA coding sequence that would make the heavy and light chain amino acid sequences of the NISTmAb, driven, of course, by an E. coli promoter. Now, those E. coli cells that made a monoclonal antibody, that they didn't make the NISTmAb, of course, I mean, not glycosylated, first of all. So they called the product of the E. coli cells making, well, they called it NISTmAb, little e for E. coli. They produced 3 different strains of NS0 cells that produce what they called NISTmAb, N for NS0. Now, these are not the same NS0 cells that make the NISTmAb. And so you could call them non-originator strains. And I will tell you that I have to be careful with the nomenclature here. But here's the point. You can engineer an NS0 cell to make, and now I'm doing air quotes, the right sequence for the NISTmAb. It's still not the NISTmAb. They also made a yeast strain of Pichia pastoris, which is now called K. fafi, that makes what they called the Y NISTmAb. All of these were used in research. There's information published about all of these, but industry basically said, no, we'd really like something where, as similar to how we use the NISTmAb when we're talking about downstream processes, we would love a CHO cell line because that's a more popular production cell line, so that we had something that makes and again, NISTmAb is not the right term here. So again, so NISTmAb is the NISTmAb. Anything that isn't NISTmAb can't be called that, okay? So the search for what would become NIST CHO was on. Some criteria were established. There were going to be some, NIST would love to release, when NIST CHO is released, it will have very few restrictions on use, but there will be some restrictions on the cells I'm talking about right now. They wanted the antibody produced by NIST CHO cells to be representative of a monoclonal antibody produced in CHO cell lines. They wanted the protein produced to have appropriate post-translational modifications and glycosylations. It was very important to industry that the cells be made in an AOF or animal origin free way. So for example, there was no serum used. And they determined that they wanted a stable construct and restricted it a little more to 1 expression cassette. So during the production of NIST CHO, there were several candidate clones, but they settled upon 1 that became known as NIST CHO. Millipore Sigma generated these cells, and at this moment in time, we do not have complete information about them. So my hand-waving explanation is what we know right now about the DNA or the expression cassette is there's some sort of promoter that works in CHO cells, driving expression of the coding sequences for the heavy and light chains that those coding sequences encode the primary amino acid sequence for the NISTmAb. That was a lot of words, and I said pretty much nothing because we don't, at this moment in time, actually know the construct. We probably will in the future, but not yet. And a very important point, even though you're going to hear me say NISTmAb for the product of the NIST CHO cells, little c for CHO, in the agreement I am supposed to say every time I talk about the product of NIST CHO cells, first of all, they don't make NISTmAb, but NIST CHO cells make NISTmAb from NIST CHO host. That's in quotes. A couple of the features, they were produced in an animal-free facility, so therefore no serum, no animal BSA, no animal insulin. There's one integrated expression cassette with one copy of the heavy and light chain. I believe that's correct. And NIST has told us that there is a PCR test looking for the border of that integration event that can easily, because it's a straightforward PCR test, that can confirm that you've got the insert. In addition, I'm going to say this in a minute, but because they're made in chosen cells, you can do that with the media as well. The CNIST.
Can I jump in for 1 second?
Yes, please.
It's a single integration site, but there are 3 cassettes in tandem.
Oh, thank you. So my slide is wrong, everybody. Thank you for saying that. Okay, so 3 copies, we think. And it has been said to me that without optimization for production, you can expect 2 to 3 grams per liter of NISTmAb from NIST CHO host. The NIST CHO cells were made on something called a chosen platform. This is a proprietary cell line that they're CHO cells with a zinc finger nuclease selection thing. It's a GS selection, glutamine, did I say that right? Synthase. And the NIST CHO cells do not require supplemental glutamine because they've already got the insert. So that's one way to keep the, what am I trying to say? So you don't get revertance, you just don't add glutamine to the media. Now, chosen cells, 1 vial, I checked yesterday, costs more than $33,000. There is a 40-page technical bulletin available online, and it's in the Slack site. And when you ask NIST for protocols, they'll say, oh, yeah, there's protocols. But really what they're talking about are the chosen cell protocols. One of the, the hackathon activity will be aimed at getting better, more specific protocols. And Maggie from MCCC and I have a nimble grant to do more of that. So the chosen cells are very expensive. The NIST CHO cells, which were derived on a chosen platform, are available right now. So the NIST CHO RM reference material 8671 is not yet available, will be released soon. The price will be announced. I'm just going to guess it's going to be about $1,000 a vial, because again, this is the recovery cost for an extensive amount of characterization and of course, creating the cells, etc. What's available right now, and why you're hearing about this, is the NIST CHO RGTM 10197. RGTM means research grade test material. This became available in January of this year. And the cost for the NIST CHO RGTM is 0. You can request the cells, you will get 1 vial of cells, you don't have to pay, and NIST even picks up the shipping cost. Leah, go ahead and ask.
Yeah, so I was just curious, it says here is CHO K1, but I think you mentioned chosen. I've heard chosen. Did they switch to a K1 for this more recent reference standard?
No, chosen is a type of CHO K1s. CHO K1s are DG44s, are several strains of CHOs that people are using. And this is a CHO K1. The reason it's called chosen is that the glutamine synthase was removed using a zinc nuclease.
Okay,
So therefore it's on the background of CHO K1, but they did some modification to it.
got it. Thank you so much for that clarification.
Thank you for asking. Okay, so who can get the RGTM? You probably can. Of course, you have to agree to the terms and conditions. This is my incredibly unofficial, shortened interpretation of the terms and conditions. According to the terms and conditions, the research grade test material is to be used to evaluate the material's fitness for purpose as a reference material. Those of us teaching bioprocessing probably want to know if it's fit for purpose for that. NIST is okay with that. I'm saying this, I am not speaking for NIST, but they seem fine with it. The NIST CHO cells are not for human or diagnostic use. I'm sure that's not a surprise to you. You must use it for non-commercial research or purposes. And just by the way, NIST asked me to tell community colleges about this. So if you're going, well, we don't really, I think they're good with it. According to the terms and conditions, anyone who gets the RGTM may not sell it or give the cells to anybody. You may not sell or give the product of the NIST CHO cells to anybody. And you may not modify the cells to produce anything other than the product it produces. Sandy, yes.
Actually, this is Maggie's question. Maggie, do you want to ask it?
Oh, the PCR test. Yeah, Zvika, can you speak to the PCR test for the insert?
And the question is, it is about whether the details for the PCR test for confirmation and detecting the inserted cassette have been published.
I don't know if it will be published, but I think that we can provide it for you. We just have to clear with Sigma. I don't think there is an issue.
Yeah, I don't
That'd
believe
be really helpful.
Just to let you know that all of this information, vector integration site, it's all going to be published. We are working on a manuscript. Well, we are in the periphery. Sigma is working on a manuscript, and they don't want to jeopardize it. Therefore, they are not divulging information, but that's all will become available in the next few months.
Good, that's great news. Thanks.
Yeah, thank you for asking that. I don't think anything has actually been published, like journal published about NIST CHO, but I know stuff is coming out. Anyway, if you break any of the rules, the agreement's void, and there is an additional condition that you have to communicate results annually to NIST. I plan to tell them that, hey, we're, whatever we're doing with them, and here's what we found. I don't think it has to be a particularly long report. How do you grow NIST CHO cells? The suggested media for the chosen cells is something called X cell media. There's more than one type of it. It costs more than $200 a liter, so it's very expensive. As I said, Maggie and I are researching using other media for NIST CHO growth and expression, and it's also one of the hackathon goals for this week. With that, I'd just like to acknowledge some of the people at NIST who have provided information, including Zvi Kelman, Brad Odell, and John Scheel. Also at MC Biotech, my institution, Collins Jones, my colleague, at MCCC, Maggie and Hettel, and I have 2 grants supporting some of this work. And with that, thank you so much for your attention. And I know that there are questions in the chat that I haven't had a chance to look at yet. Sam?